Hello students, this is Geethanjali. In this video, we'll be learning about the tRNA molecule. So tRNA molecule is also called the adapter molecule because it is able to read the genetic code from mRNA and help in the formation of protein. And it is also called the soluble RNA because it is soluble in sodium chloride solution. RNA, like proteins, has three structures. It has primary structure, secondary structure, and tertiary structure. So primary structure is just a sequence of nucleotides, whereas secondary structure, you can see here, this is your secondary structure and it is called the clover leaf structure of RNA, tRNA. In this structure, there are four arms, you will notice. So what are these arms? Arms are basically those places where there are no hydrogen bonds. Okay, these white marks that you can see denote the hydrogen bond. So wherever there's no hydrogen bond, those places are referred to bonds. That is why you can see the molecule is kind of separated rather than joined like in these cases, right? So in this secondary structure of tRNA molecule, we'll see the 5' end starts here and it moves till the 3' end. So the amino acid which is there, as we discussed the function of tRNA, it basically reads the mRNA code and it brings the required amino acid to the ribosomal complex during protein synthesis for the formation of the protein. So the amino acid which is there, it is attached to the 3' end of the tRNA. And there are 61 types of tRNA as there are 61 types of codon. Whereas the three other stop codons, they do not have tRNA. Now let us assume this is our mRNA and this mRNA has the genetic code, or let us say it has all the codons over here. So the tRNA which is there, in one of its loops, or rather we would say arms, there's a part which has the anticodon. So by looking at the anticodon, we can identify which amino acid it is assigned for. For example, here there is AUG, okay? The corresponding code will be USA. So in case we see a anticodon, we call this anticodon because here we have our codon on mRNA, so we'll have anticodon in the tRNA. So in case the anticodon is UAC, we can easily identify by just writing the corresponding, the complementary base. So we have AUG and we know that AUG codes for methionine. So we know that the amino acid that is present here is, that it carries is methionine. So like this 61 types of RNAs with different 61 codons are there for amino acids. As I told you, it also has tertiary structure. This tertiary structure is relatively more complex than the secondary structure, and it is like an inverted L. Okay, so it is quite complex and cannot actually be depicted on a 2D surface like how I'm showing you, but It is said to have an inverted L shape. So these were a few points about tRNA.